conservation of sea otters. So thank you, Chanel. I knew I could count on you.
What are you trying to get?
So Shawn, you've got the floor. It's all yours.
Thanks, Bob and Chanel, and thank you for inviting me to speak today. All right, I'm going to start sharing my screen. Give me a moment here. And then, okay, can you see my PowerPoint?
Yes.
You're good.
All right, thank you. Thank you again for having me today, and thank you for everyone for attending this science symposium. Elakha always puts on great events, and this is one of them. I'm going to talk about the historical and genetic considerations of Oregon sea otters and sea otters in general. This is chapter 4 of the feasibility study, which was written by myself, and I'm Dr. Shawn Larson, Curator of Conservation Research at the Seattle Aquarium. I have been there since 1995, as Bob said, and Dr. Tim Tinker, who is CEO or President of Enhydra Ecological Consulting and an adjunct professor at UC Santa Cruz. So starting out, just wanted to point out the historical and current range of sea otters. I know that if you've been to this symposium, you have seen this map before, but this is a historic and current range map. Sea otters historically occupied the areas in the pink shading to my eyes, and then gray. So all the way from northern Japan, along the Kamchatka Peninsula, through the Kuril Islands, all along the Aleutian chain, the Alaska Peninsula, central Alaska, and then all along the west coast of the United States and Canada or North America, down to the center of central area of Baja. Prior to widespread extirpation during the fur trade, which ran from the mid-1700s to about 1910. Prior to that, there were an estimated 150,000 to 300,000 sea otters throughout this area, and it is a very wide estimate there because nobody was really counting sea otters back then. And then after the fur trade extirpations, 99% of the sea otters were wiped out, leaving these remnant populations widely scattered. There's 11 of them that survived the fur trade, and they are the populations that were left very small, about 1,000 throughout this whole area. So each population had anywhere from 10 to 100 individuals, or maybe a little bit more than 100 individuals. But these are the populations that were left after the fur trade ended in 1910 to recover sea otters. And they did recover sea otters throughout much of central Alaska, the Aleutian Islands, Kamchatka Peninsula, and the Kurils, and then this little area here in central California. But then you see there's otters here in the purple or pink, and those are the otters that are represented by the reintroductions or translocations that occurred in the 60s and 70s that Jim Bodkin talked about the first day of the symposium. And this is why we have sea otters today in this area in Southeast Alaska, basically from the edge of Prince William Sound all the way down, almost continuous to central Washington. But there still remains this big gap of previously occupied territory here between central Washington and central California. And this is why we're interested in new reintroductions today to fill in this gap. The current population estimate of sea otters today is, this is an older estimate that was from our 2015 sea otter conservation book. At that time, it was 125,000 sea otters. Is probably well over that. Not exactly sure. We haven't had a really good range-wide count recently. So I'd like to speak a little bit about First Nations people and their relationships to sea otters. First Nations people have had a direct relationship with sea otters well before the fur trade for thousands of years and potentially tens of thousands of years. And I have a couple of quotes I would like to read. The source is Solomon et al. In the Sea Otter Conservation book, her chapter was First Nations Perspectives on Sea Otter Conservation in British Columbia and Alaska, Insights into Coupled Human Ocean Systems. And I will not try and pronounce the author of this quote because I want to make sure that I get the pronouncement right, and I'm not sure that I can. But at any rate, the quote is, "People have to understand how valuable the sea otter is to our people. We have great histories. We have been with them for years and years, thousands of years. Big chiefs use sea otters to recognize a great chief amongst our people. The sea otter can bring back all the histories of people before." And the images here are, this one is of a whale fin made by the macaw out of cedar. And it's an piece of art. And it is inlaid with over 700 sea otter teeth. So First Nations people were using sea otters for their pelts here. These are two elders wearing ceremonial sea otter gowns, and they also used their teeth for decorations. And then they also depicted sea otters in many art pieces. And this is a Tlingit drum, and is one of my favorite First Nations pieces of art that depicts sea otters. And there is more and more evidence of the hunting practices that First Nations people use for thousands of years when working with sea otters. And there's evidence for spatial management and exclusion from just not all areas that sea otters occupied, but only certain areas. And here is another quote from a First Nations chief, Dominic Andrews, and this is also from Solomon et al. In 2015. And the quote is, "In our history, certain people had rights to certain areas of Hesquat Harbor. They had licenses to take sea otters for harvesting. Sea otters were hunted at certain times of the year, not every month. Certain people had rights to the pelts. The pelts were distributed to other First Nations for their regalia. These were people with hierarchy. So the sea otters were highly prized for their pelts, but they were also prized for their role in the ecosystem. And they were excluded in only certain areas. And these are the areas that the First Nations people would exclude the sea otter from so that they could harvest the large benthic invertebrates that people like to eat. The sea otters also like to eat, like clams, crabs, urchins, etc. So they would exclude sea otters from certain areas to preserve their clam gardens. And they allowed some populations to remain locally and did not systematically wipe out all populations as the fur trade, thus preventing widespread extirpation of sea otters. There is genetic evidence, however, that First Nation hunting did cause some population bottlenecks, resulting in lower diversity. And this is some work that was published by Aguilar et al. 2008 and Beichman et al. 2019. And I also have some evidence that shows that there were population bottlenecks affecting genetic diversity prior to the fur trade, but the fur trade also had a big impact on population diversity. So let's talk a little bit about the historical otters in Oregon. The sea otter remains one of the most common marine mammal bones found in First Nations midden remains all along the Oregon coast. And middens are collections of shells and bones left over from First Nation occupations in their original tribal areas. And this is a map of Oregon, and it shows you the locations of First Nation middens that have sea otter bones in them, as indicated by these dots all along here. So this is Northern Oregon, this is Central Oregon, and this is Southern Oregon. And this map is from Karan, Cohn, and Wickezer, 2019. There are also written accounts of sea otters occurring in Oregon from fur traders. And in fact, the Lewis and Clark expedition in the winter of 05 and 06 that they spent near the Oregon coast described sea otters as plentiful but hard to kill. There are also some historical accounts from the fur traders that pointed to actually few fur trading ships that operated out of Oregon, probably because there weren't many ports that they could easily get in and out of. But yet, even though there weren't a lot of fur trading ships working out of Oregon, the original Oregon sea otter was hunted to extinction during the fur trade, and we think likely by local hunting, but also assisted by fur traders coming from other areas. So sea otters definitely occurred in Oregon before the fur trade, as evidenced by First Nations midden remains, the bones that are there, as well as oral histories from both First Nations and fur traders and Lewis and Clark. So looking at genetic studies of historical Oregon otters, there have been a few. The first one was done by Valentine et al. 2008, and she looked at mitochondrial DNA, a portion of the mitochondrial DNA, which is inherited from the female only. She sequenced, I believe it was a 400 base pair segment, and from these two areas in Oregon, there was this one site here in North Central Oregon and a site in Southern Oregon. And she found the dominant mitochondrial type or haplotype matched the current Southern sea otter. So she concluded that the otters that used to live in Oregon in those areas were the Southern sea otter. And I did some work that I published in 2012 that looked at nuclear variable markers or microsatellites. And I took original sea otter bones from 5 sites here from North Central Oregon, Central Oregon, and Southern Oregon. And I found genetic signatures that match both Northern and Southern sea otters. And the Northern sea otter signatures I found were the ones in North Central Oregon, and the Southern sea otter signatures I found were the ones from more Southern Oregon. And then Wellman et al. Most recently published in 2020 her work, and she completely sequenced the mitochondrial genome. She had 10 individuals from 2 sites in Northern Oregon, and she found that those mitochondrial types most closely match the Northern sea otters. So all 3 of these suggest that there were both Northern sea otters and Southern sea otter genetic types in Oregon originally. And we think that there was a genetic latitudinal cline throughout the state with a hybrid zone between the 2 recognized subspecies, the Northern and Southern sea otters. Okay, genetic considerations for reintroductions. We want the genetics to be consistent with the original Oregon sea otters, which we now think are both combination of both Northern and Southern sea otters. We would like to recover genetic diversity because sea otters did lose significant genetic diversity during this fur trade extirpation as well as earlier extirpations. Again, 99% of the otters were hunted for their fur, and they did lose genetic diversity. And then we want to recover the genetic connectivity because we still have this big area here that it remains unoccupied. Okay, so let's talk a little bit about the genetics of successful reintroductions. They have recovered genetic diversity and they have restored connectivity or gene flow between populations. Okay, so I did a study that was just published this year, and I have tissues from all 3 recognized subspecies of Bering Island, which is the Northern sea otter or Russian sea otter. I've got multiple samples here from the Northern sea otter or Alaskan sea otter from the Alaska Peninsula, Katmai, Prince William Sound, as well as some of the translocated or reintroduced groups. So I have samples from Southeast Alaska, BC, and Washington, as well as California. And here are the sample sizes. And again, wanted to point out to remind people from Jim's presentation of the source population. So we have the Southeast Alaska population that was primarily founded by Amchitka animals and some Prince William Sound animals. And then The Vancouver Island population was a third founded from Amchitka, 2/3 founded from Prince William Sound, and then Washington that was exclusively founded from Amchitka animals. And Amchitka is an island way out here. And then here's Prince William Sound. So they took animals from Amchitka and Prince William Sound and brought them to Southeast Alaska, brought both of those source populations to BC, but only animals from Amchitka came to Washington. Okay, so let's talk about how it has recovered genetic diversity, those reintroductions. So these are the genetic diversity estimates for the average of all the remnants. And so from Bering, Alaska Peninsula, Katmai, Prince William Sound, and California, and you can see that they have relatively low genetic diversity. These should be in the 70s and 80s. They're still in the 50s. So that's from the remnants. And then when you look at the averages of the reintroduced, they're higher. They're not significantly higher, but they're definitely higher. And when I did some work on pre-fur trade otters, bones that go back anywhere from 1,000 to 10,000 years prior, we did find genetic diversity estimates between the mid 60s to the high 70s, which is what is expected for mammal populations that have not been through a significant bottleneck. But what I wanted to point out is the Southeast Alaska diversity has the highest genetic diversity of any of the populations measured. And they're approaching that lower level of the pre-fur trade genetic diversity, which is great. And we think this is because the Southeast was founded by two genetically distinct populations. So from Amchitka and Prince William Sound. And then here we've got Washington and BC, which actually BC levels of diversity look very similar to their the most numerous founder, which was Prince William Sound. And then Washington is higher than that and is approaching the levels of Southeast Alaska. So that's good news there. If we look at population differences and similarities, this is a program called Structure that uses the genetic data to determine the most likely population structure given the data. And so when it looks at just the subspecies differences, it pulls out California as distinct and does not recognize Bering as a distinct population. So Bering is being clumped, or the Russian sea otter is being clumped with the northern sea otter, but California is distinct. When you ask the program to consider finer population structure, what happens? So this is 3 different populations as indicated by the 3 different colors. You see that Prince William Sound also seems to be genetically distinct. And then when you ask the program to look into further genetic structuring, then it pulls out Prince William Sound, California, and then clumps the reintroduced populations together. So BC, Southeast Alaska, and Washington are grouped together, which makes sense because they all have common founders. And then Alaska Peninsula, Bering, and Katmai are grouped similarly, have a similar dominant color. And this makes sense because they are all connected. Katmai and Alaska Peninsula are at the eastern edge of the Aleutian chain, and then Bering's at the other end. And since there are populations all in between there, we think there was likely gene flow between all of those populations, although we just have samples from Katmai and the Alaska Peninsula, and then Bering, we don't have all those other populations intermediate to that. But what I find fascinating is with all of these different iterations, Bering is never pulled out as a distinct genetic population, even though it is recognized as a different subspecies. So what this is really pointing to is the genetic structure is pointing to stocks, not necessarily subspecies. And this is another program called TESS, which has a different algorithm to group distinct populations based on the genetic signatures, and the colors are showing you the most likely stock structure that this data set is pointing to, and it shows California as distinct. Again, all the reintroduced populations which have common founders as distinct. Prince William Sound is distinct, and then Katmai, Alaska Peninsula, and the Bering population is also similar and distinct. When we look at subspecies differences, looking at mitochondrial DNA, which I'm not presenting here, there is a less than 1% genetic difference between the subspecies, which is relatively low for a marine mammal. It's lower than most of the other marine mammal subspecies differences. So I don't want to talk about subspecies validity with genetics here, but there certainly maybe should be some more work looking into whether or not the Northern and the Russian are distinct. California is definitely distinct, but then even with that, there's some low differences between them all, which is good news for future reintroductions, looking at bringing some of the Alaska animals down to Oregon, potentially, and California up to Oregon. There's really not that much genetic difference between the groups. Okay, then talking about gene flow. So we actually ran this data set through 3 different programs to determine gene flow. This was the most conservative. And what it showed is that there's a lot of gene flow between many of these adjacent populations, for example, between southeastern BC, the Alaska Peninsula and Katmai, southeast in Washington, Prince William Sound and southeast, and BC and Washington, which makes sense. But really all of the data is suggesting that there's gene flow between all of the sample populations, except for those that were the farthest away, which was California and Bering Island. Okay, getting back to the map here. So what we found is that there's really genetic connectivity or gene flow as a result of these successful reintroductions, really bridged the gap here. And now we've got gene flow between most of this area as indicated by this study, which is really great news for the recovery of sea otters, especially genetically, but we still have this really big gap here between South Central Washington and Central California. And that's why we're discussing reintroductions. One of the reasons. So genetic considerations for reintroductions, diversity. We want diversity maintained, and the previous successful reintroductions actually maintain genetic diversity even with a small sample size, small founding size. And the highest diversity was measured in the reintroduced groups with 2 or more founders. Then the gene flow piece, the successful reintroductions resulted in increased genetic connectivity between groups, essentially making all but Bering and California connected, which is really great for the period of 50 years where, from the point where these populations were reintroduced to where they are today. So potential source populations, genetic perspectives for Oregon. Historically, again, Oregon had genetic signatures of both southern and northern sea otters. So how do we recreate this? We can have 2 separate sources for reintroductions. There can be northern sea otters released to a northern Oregon site and southern sea otters released to a southern Oregon site. And then over a shorter period of time, these 2 would merge, hopefully. Or could release northern sea otters into Oregon release sites and then southern sea otters to Northern California release sites, and mixing will eventually occur naturally. Or there can be just 1 source for reintroductions, and potentially southern sea otters might be better to move from California into Oregon from a genetic perspective, because we already have northern sea otters in central Washington, and we're hoping that they will move south quickly. Well, we don't know, sea otter range expansion is not very quick, but mixing will eventually occur. So there's a few different scenarios there, and it's not for me to point out which one is best. That's for somebody else. At any rate, just wanted to share with you a raft of reintroduced Washington sea otters at North Rock, just west of Hohead. This was, I think, in 2015. This raft, which is in central Washington, and that's cell phone video through a spotting scope, is about 500 animals. So this is what you want to see in Oregon, and this is what we're seeing pretty consistently now in Washington, especially the southern areas of the range south of La Push. So we were really thrilled to find this in our spotting scope. We weren't expecting it, This raft of sea otters looks like kelp. Yes, these are all the sea otters here. But when you zoom in on it with a spotting scope, you can see all of them there. And this is just a closer look of some Washington sea otters just lounging around in the near shore. And these guys do haul out. And they're very big and fat and happy and rest a lot. And so they're doing really great in Washington. And I think that they will do great in Oregon as well. And that's all I have. If anybody has any questions, here's my email. And that's it. I'm happy to take questions now or at the end of today's session.